My name is Peter Cooper. Both Tao and I are members of the NCBI service desk, and we're two of the people that answer BLAST help. So this talk today is going to be called A Practical Guide to NCBI BLAST. We're going to be focusing today on the web interface, and we're going to be dealing with the kinds of problems that we know people have with that interface because we get a lot of questions in the BLAST help email alias. And when doing some exercises at the end of it, you'll need to use sometimes the saved BLAST request IDs, and I have a document that has those in there. And those will be valid, many of them, for about a month or so. So if you want to go back and review some of this stuff, if you follow that materials link that's on this slide, and we'll make that available after the webinar as well, you can probably reproduce some of the examples. So we're going to talk a little bit about what BLAST does. Why would anybody use BLAST? BLAST statistics, what they mean, BLAST scoring, how it works, and the various search programs that we have. And then we're going to focus a lot of the slides we'll be talking about practicalities of using the web interface, some things that you need to know about that we recommend you use. And then we're going to talk about some of the other services that are extensions of BLAST, Cobalt, which is our protein multiple alignment tool, Primer Blast, which is our primer designing software, and a relatively new clustering tool called Mole Blast. And once I get through the slides, we'll go ahead and do some live searches using BLAST itself and these extensions of BLAST. Okay, so just a little bit of a definition. BLAST stands for Basic Local Alignment Search Tool. It's probably the most widely used sequence similarity search tool, especially when you're talking about large database searches. What it does is it finds these things called high-scoring local alignments between two sequences. And you can have protein sequences or DNA sequences. And as we'll talk about in a few minutes, you can also translate DNA sequences. BLAST includes a model of score distributions for random local alignments. And so because of that, we can tell if our alignments are different than what we would expect by chance. So it provides some kind of statistical significance for our alignments. Let me come back to that point in just a second. So what BLAST really tells you is about non-chance similarities between biological sequences. And that's an important thing for biologists because if the similarities are not due to chance, then they must be due to something else. That something else is typically thought to be homology. That is that those two genes, those two proteins, those two transcripts are descended from some common ancestor. So that's the concept of homology. Probably the most common use of BLAST, however, is just simple identification. Does this sequence exist in the database? In order to use BLAST, you have to have a sequence, either a protein sequence or a nucleotide sequence. It could be one that you determined yourself, or it could be one that you've taken from the NCBI databases. So one way of thinking about what BLAST tells you are the kinds of questions that people are asking. The first question is sort of the one that BLAST was intended to do in the first place, and that is, what's my sequence related to? And a related question to that is really the one that people usually want to know when they ask that question, is what does my protein sequence do? So that's about homology, what's it related to. And it's also about function. Homology does not necessarily mean that the two things have the same function, but we often make that leap, and there's a kind of BLAST called Rps Blast, or conserved domain database search, that might be a little bit more informative when we're thinking about function. The second kind of question is this identification question. Does this sequence already exist in the database? Find the matching sequence in the database. And a lot of times what we're thinking about these days when we're doing that, especially with the advent of a lot of the metagenomic sequences, like the gut metagenome sequences and things like that, what's the organism of origin? And then finally, the third question is, where is it located, or how is it organized? Where is it in the genome? If I have a messenger RNA sequence, it's a transcript of a gene, where's the corresponding gene? And I also can use BLAST to look at other annotation problems. I have a sequence, there might be something wrong with it, an error, either one that's a sequencing error, or there's something bad with the sequence. This is something from nature that has a frame shift in it or something like that. It's a mutation. So as I said, BLAST has a statistical model for how random sequence alignments behave. And the important statistic that you get from BLAST is something called the expect value, or the E value. The expect value is the expected number of random alignments with a particular score or a better score. And so if that number is large, it means that I can't distinguish my alignments from chance. You can see the alignment at the top of this slide. That's kind of a standing joke around here and everywhere. You can find Elvis in the sequence database, but this peptide with five amino acids occurs by chance in a lot of cases. So in this case, the expect value for Elvis is about 48,000. So I would expect 48,000 alignments with a score of 36 or better. So I can't tell you anything about Elvis. Is it due to chance? I don't know. I can't tell. And that's what that means. The alignment at the bottom has an expect value that reads 7e minus 18. That's just our way of expressing 7 times 10 to the minus 18. So that's the number of chance alignments that we would expect. That's essentially 0. So this alignment and the score that comes from it is not due to chance. A couple of points about the e-value that are important when you're thinking about blast searches. The e-value depends directly on the size of the search space, and generally the size of the search space is governed largely by the size of the database. So it's important to search the smallest database that's likely to contain the sequence of interest. We'll come back to that point later because it's very important when you do blast searches on the web to sort of restrict your database a little bit so that it's not so big. How does BLAST score alignments? The simplest scoring system is the one that's used for nucleotides, where we have a positive score for a match, a negative score for a mismatch. We have some kind of gap cost. This is the way the gap costs are calculated for BLAST-N. They're slightly different in the way they're calculated for another algorithm called MegaBLAST. But the nucleotide algorithm is not terribly sophisticated in terms of its scoring system. And then you can see at the top, you'll get your score. There's a bit score and there's a raw score. But the important statistic that you can learn something about is the e-value. So it tells you that this alignment is not due to chance. The protein scoring system is more sophisticated. It has a concept of conservative substitutions built into it. The scoring system we use is something called BLOSSOM 62. And I could pull that matrix up through that link on the slide. I'll let you do that on your own. But basically what we see is that some amino acids are more important than others. They're worth more in terms of a positive score. And so we have this concept of conservative substitutions where things like glutamate and aspartate, the D and the E there, they still get a positive score. Other kinds of alignments like a glutamine for phenylalanine, they get a negative score, and so on and so forth. Those scores are what are called log odds scores, and they essentially tell you whether the particular substitution occurs more often than we would expect by chance or less often than we would expect by chance. And then we have a gapping system just like we had for the nucleotides. In this case, we have an alignment between two proteins. They're actually a mammalian and a bacterial globin proteins. The expect value is 4 times 10 to the minus 50th. That means that we would not expect an alignment score this good by chance. So let's deal a little bit more with BLAST. And I'm going to be talking somewhat in the abstract here because these things are available as standalone programs. Of course, today we're talking about the web. So we have these nucleotide search programs. There are 2 of them really. BLAST-N, which is traditional BLAST algorithm. This is the most sensitive kind of nucleotide search you can do on the web. There's another algorithm called MegaBLAST that is a slightly different algorithm. It's not technically BLAST. It has a couple of things about it. It uses a larger word size than BLAST-N, which means it's more likely to miss things. It has a slightly different gapping model. It comes in 2 flavors, contiguous MegaBLAST and discontiguous MegaBLAST. Contiguous MegaBLAST is the one that's turned on by default on the nucleotide search pages, and it's the one that's best for identification or same species annotation. In other words, where is this gene in my genome? And that's what NCBI uses that algorithm for. We do a lot of our annotation with MegaBLAST. So when we come back, when we look at the web pages in a few minutes, you'll see how these things are organized and how you pick the one that you want. Protein search programs really come in 2 varieties in terms of the kinds of scoring matrices that they use. The traditional BLAST ones use what are called position-independent scoring, and I should have pointed this out when I showed you the slide that had the BLOSSOM 62. A substitution in BLOSSOM 62, the same substitution in an alignment gets the same score no matter where it occurs. And that's not a terribly realistic model for proteins. We'll come back to a more realistic model in just a minute. So the BLAST-P is a traditional protein search algorithm. There are also translating searches. Now, you might find it strange that I call these protein search programs, but they are, because the actual searches themselves are at the protein level. There are 3 different ones: BLASTX, where you can translate your query sequence, TBLASTN, where you can translate your database, and TBLASTX, where you can translate both the query sequence and the database sequence. And these do 6-frame translations, and they're very useful for unannotated protein coding regions on nucleotide sequences. Now, the other kind of program that I want to mention, and we're not going to spend any time doing searches with these today, except for the one that we get by default, and I'll come back to that in a minute. These are BLAST algorithms that take into account things that we might think of as protein domains, and they use a position-specific scoring model. Position-specific scoring is based on some kind of a multiple alignment of proteins that we know are related to each other, and the positions in that alignment. What we observe, of course, is that the substitution scores depend on the position in the alignment. That's because some places in the protein are more important than others, and they're less likely to change. These kinds of scoring systems give much more accurate alignments, and they are more sensitive at identifying distant homologies. They also are the way that we go about identifying conserved structural and functional domains in proteins with our conserved domain database search. So here are the position-specific programs that we have available. And like I said, I'm not going to have time to talk about these today, except for we'll see the one in action. That's reverse Sra Blast. This is called Rps Blast. And this runs with all BLAST piece searches at the NCBI. And it's going to search a database of Sra Blast generated position-specific score matrices. It searches our database essentially of conserved domains, and it quickly identifies basically what kind of protein is this, and potentially gives you access to a function and perhaps a structure for that kind of protein. All right, so as I said, today we're going to be looking at accessing BLAST on the web. And from our homepage, which is in the process of being updated soon, but as of now, you can get there quickly from the BLAST link. And of course, it's very easy to find BLAST. If you use Google, you can get straight to the BLAST homepage that way. And here's the BLAST homepage. It's organized into 3 sections, basically. It has its own sort of unique URL. Instead of being a www URL, it starts with a BLAST. So it's blast.ncbi.nlm.nih.gov. The 3 sections, we'll talk about these in sort of a mixed way today, because really I want to point out to you that some parts of this are just separate interfaces to the same databases. In particular, we're talking about the assembled genomes and things like that. So we have a way of getting at the assembled genomes with a fairly new autocomplete search box up there that's really a good way to find data. We have the central part of the page, which we're calling Basic BLAST. These search the traditional BLAST databases using the algorithms that we talked about. And then at the bottom of the page, there's sort of a mixture of different kinds of things that we're calling specialized BLAST. Some of these things are not really BLAST. They use parts of BLAST. Some of them are interfaces to special BLAST databases. And we'll talk about these, the ones I talked about earlier today. We're going to talk about Primer BLAST, Mole BLAST, and Cobalt. So if you're coming to BLAST, the first thing you're going to have to do is come up with a query sequence and input it somehow into the box that's on the BLAST form. This slide just points out to you the kinds of things you can do. Number one, I just want to point out to you that you can put more than one sequence in there, and we'll come back to that point in a few minutes. So you could put a whole series of FASTA formatted, in this case, nucleotide sequences there on the left. You can also enter accession numbers or GI numbers, other identifiers from NCBI, one on each line. And there's actually a separate interface for those that you can take sequences that you found in, say, a sequence search at the protein database or the nucleotide database at NCBI. Just click this link that says Run BLAST, and that will throw them into the query box. Another point, a related point, and this is listed as a separate service down at the bottom of the BLAST homepage, but really it's not anything special. On any BLAST form, you can check this little box that says Align 2 or more sequences, circled here. And when you do that, the interface changes. It opens up a separate box in the bottom. And so you can take the bottom box and you can generate your own little database, a small custom database, or you can just use 2 sequences. The original intention was 2 sequences. Or you can do a small all against all with your own sequences. One of the points I want to make here is that many people who use this service called BLAST2 sequences are really not interested in a local alignment. They're interested in what's called a global alignment. A local alignment like BLAST creates is only going to show you the high scoring parts of the alignment. That's why it can give you some statistical properties about your alignment. Many people just want to get a global score from this, like a % identity or something like that. We do have a tool for doing that, and it's listed at the bottom of the page, but I thought I would bring it in here. And that's the global alignment tool, which is our sort of implementation of something called the Needleman-Wunsch algorithm. And I've shown you the link here. It's on the bottom of the BLAST homepage, and it basically will do a pairwise global alignment. It includes all residues of both sequences. And one point to make about global alignment tools like this one is that it will align unrelated sequences. It will align anything to anything, so it matters a lot that what you give it are two sequences that are actually related to each other. It does provide the global statistics, the % identity, and the positives. Those, by the way, are also shown to you in BLAST, but they don't mean the thing that you think they mean there. They're only referring to the local alignment. This alignment here is the alignment between albumin and a protein called the vitamin D binding protein. Albumin has a repeated domain structure. The vitamin D binding protein is missing one of those domains, and that's why we have this big gappy region in the middle. Is this a biologically realistic alignment? Maybe not. And that's one of the things to be careful about when you use global alignment tools like that. Now, I've talked about using multiple sequences on the web in BLAST, and I just want to point out to you that many people write to us and they want to know, look, how many sequences can I stick in that box and realistically expect it to work? And there isn't a good answer to that question. You will occasionally run into this red warning that's shown on this slide here, and it's a little cryptic. It asks you to write to us and include the RID so we can take a look and see what happened. But that message that says CPU usage limit was exceeded tells you that you sent too many sequences to us. We don't have a fixed limit on the size or number of queries, but we do limit it by processing time. So you have about 1 hour of CPU time for a single search. 1 hour of CPU time is not 1 hour of real time because there are multiple CPUs involved in running BLAST search. Can you estimate the processing time? Not really, because it depends on a lot of things that depend on the database and the nature of your query sequence. So the size and number of queries, of course, is important, but the nature of them. How many hits did they get back? How big was your database that you searched? So keep it at a modest level. If you need to have large access to BLAST, high volume access to BLAST, that's a topic for a different webinar. We have a BLAST in the cloud service and other things like that. We're talking about using our web service here today. Okay, now this I think is one of the thorniest things in terms of using BLAST, is knowing which database to search to find your sequence of interest. And there are two main sets of databases on the basic BLAST pages. We have a set of protein databases, and of course we have a set of nucleotide databases. So this is what the protein database pull-down list looks like. There is a default database, and most people who use BLAST are familiar with the concept of the NR database. This is our non-redundant protein database. It is a fairly good choice if you want to search a fairly large set of sequences that's fairly comprehensive. It also has some useful subsets, our reference sequences, the sequences from the SwissProt part of UniProt, and PDB, the sequences from structure. Those are all useful to search separately. They provide specific kinds of information, but those data are included in NR. The question about what is not in NR is important. So there are basically three things that are not present in the NR database. Those are the patent sequences. These are coming from US, European, and Asian patents, and we get them in a passive way from the European and Asian patent offices because they submit those sequences to their corresponding sequence databases. The US patents come to us automatically, so we make no assertions about the completeness of that data or even its accuracy, but they're there in case you're interested in searching them. Also, a separate set of data are the proteins that have been made from metagenomic sequences. These tend to be a couple of studies. This is not all of the metagenomes, but it's some of the early studies, Craig Venter's Sargasso sea genome and things like that. And then there are proteins from next-gen assemblies. These are from our Transcriptome shotgun assembly database. This particular set of data on the nucleotide side is more important than it is for now on the protein side. And so that brings me to talking about the nucleotide databases, which are much more complicated in terms of knowing which one to search. And one of the big problems here is that the default database, which is also confusingly named NR, I like to think of it as NT just so we can distinguish it from NR, is really not the master of everything else. So there are lots of big sets of data that are not included in NR. And I want to come back to that point right now. So as I said, the default database is not comprehensive. What's in it are traditional Genbank sequences. These are things that meet the standards of going into the old-fashioned Genbank divisions where they have the primate division and the plant division and so on and so forth. There's a certain standard of accuracy. And originally, there was a certain idea that they would be well-annotated sequences with lots of biological information, plus our reference sequence RNA sequences, which are a very useful set of data. You can also search some of these things separately. You can search the RefSeq RNA, and you can search the 16S RNA reference sequences as separate databases. Those are both useful sets. And in fact, we'll do a search against the 16S rRNA sequences later on. We'll do that using Blast. So it's important to keep in mind what is not in the default nucleotide database. And the answer to that is the majority of the nucleotide data of NCBI is not there. Bulk sequence divisions from Genbank, the ESTs, the GSSs, the HGGSs, and the STSs, they're not there. They tend to be of lesser importance these days as most people are not actively producing these kinds of sequences anymore. The RefSeq genomic sequences are in two different databases, Chromosome for complete chromosome records and RefSeq genomic for large contig sequences or assembled contig sequences. These are not in NR. And of course, the European and Asian and US patents are not in there. And then there are two very large sets of data that are not present in our whole genome shotgun contigs. These are the unassembled whole genome shotgun contigs. Many of these data are available elsewhere, in particular through the Genome Blast pages. The next-gen reads from the SRA database. This is the largest set of nucleotide data at NCBI. These data are not available in the default nucleotide database. We recently added this as a database on the pull-down list for the nucleotide pages. We're going to use a separate interface to this because you really need to know what set of data you're searching. And then finally, the transcriptome shotgun assemblies. This is a very large set of data that's growing rapidly. These are assemblies of next-gen sequences into cDNAs in particular, but they're not very well annotated. So they're not included in R, but they're useful to search, especially if you're working with an organism that doesn't really have a lot of support at the traditional Genbank level. Now, I said when we're talking about databases, the most important thing that you can do is to search the smallest database that's likely to contain a sequence of interest. That means that you really should never have to search NR. It's very unlikely that you really need to cast that wide of a net. So there are lots of things you can do, and this modifies your search ahead of time. So the most important thing and the most useful thing is to use some kind of an organism limit. Here's an example. I said, give me all the bacterial proteins, but get rid of all this order that's overrepresented, for example. Say I'm not interested in enterobacteria, I can get rid of that. I can also exclude certain kinds of sequences. If it's a bacterial search, I might not want to see any of the uncultured bacilli or things like that. And then I can use an Entree query. Any Entree query that's valid in the sequence databases, nucleotide or protein, depending upon which page you're working with, you can use that to further limit your database. So this is an important thing to do almost every time you run a blast search. Now, I mentioned the genome databases a minute ago when I was talking about the whole genome shotgun contigs. This is a better way to search genomic data, is to use this selector that's on the home page, because it will get you to the page that gives you the most assembled set of genomic sequences. So there may be a bunch of whole genome shotgun contigs that are sort of loose in the database in WGS, but sometimes they've been assembled into a larger record like a scaffold or a chromosome, and that's probably what you want to search. So go here if you're interested in doing that kind of search. So in a lot of ways, this is just another interface to getting at that data, and that will take you to some BLAST pages that are set up specifically to search this kind of data. We're very busy annotating eukaryotic genomes. So for example, the top page here on the left, if you go to the white-throated sparrow page, you're going to get the reference assembly, and these are basically the chromosomes of that organism. This is the one we'll actually use today, the chicken gut metagenome WGS project. You can also find that in the organism selector there on the genome page. These are going to be contigs, but still, you can search them directly using this interface. It's a lot easier than searching the WGS database on the NCBI basic BLAST page. And then SRA, as I mentioned to you, SRA is available on the pull-down list on the nucleotide pages. But really a better way to get into that is through the interface, the SRA interface itself. So I can go to the SRA Entree database. I can find the experiments that I want, select them, and then send them to BLAST. And what that basically does is it sets up a page for me that has those experiments. In this case, this is a fossil bone, horse bone from Canada, and I could search against that. In this case, I'm searching with the horse mitochondrial genome. But there are all kinds of studies in there. We'll look at one today when we do an SRA BLAST. Just a couple of points that we talked about programs. We do need to select our program on the web. This comes up more frequently for the nucleotide searches because this is where you have to decide whether you're going to use MegaBLAST or BLASTN. And so on the web form, you can pick your program. By default, it's going to be MegaBLAST. If you're trying to look at slightly more distantly related organisms, you may need to change this to BLASTN to get a more sensitive search. And hopefully we'll see an example today where you can see what kind of difference it makes when we do a cross-species comparison of two plants. Protein page, the only choices here are whether you want to launch into one of the position-specific searches other than conserved domain search. That's run by default. If you want to run something that's going to generate your own position-specific score matrix, you would have to select that here. You can always launch into any of these programs using the formatting options on the Protein BLAST results. Now there is a section of the page that is hidden away at the bottom of the form. It's called the algorithm parameters. And we hid it down there because we didn't think you would need to adjust things down there, but it's actually not true. I want to point out there's a couple of things down there that you really need to keep in mind. And the first one is the general parameters section, and this governs your output. And it's very important that you stick to a rule. The rule is that you want your output to be governed by the expect threshold. Notice that the expect threshold is set to 10, so that means you're going to see hits that are as bad as an expect value of 10. Remember, those are indistinguishable from chance. So it's pretty important here to set that to a more stringent value. Here are some examples of more stringent values. This depends largely on what your goal is and what your tastes are as to what's a stringent value. But you can write them just like BLAST writes its expect values. The other thing you probably want to do to make sure that this governs your output is to change the number of max target sequences. So it's set to 100 by default. Typically in a protein BLAST search, unless you have extremely limited your database, you're going to need to set this to 500 or higher. So just keep that in mind. If you don't reach your expect threshold cutoff in your output, go back and run your search again and change the number of max target sequences. If you're working with nucleotide data, especially if you're working with genomic data, it's real important to pick a species-specific repeat filter. This filters the things that are in eukaryotic genomes that are called interspersed repetitive elements like ALU or line sequences. So here's a simple example. This is the BLAST graphic. On the left-hand side, the repeat filter was not on, and you can see all those hits in that one area. That's where there is an L1 element in this particular sequence. If I turn the repeat filter off, then I don't get those, and I get the hits that I'm looking for. So keep that in mind when you're working with this. This is on by default on the genome-specific BLAST pages for obvious reasons. Okay, then once you get your results, there's some things to keep in mind, and one is the ability to format your results in different ways. And this is managed through the RID. You can just go back to the web server and get back your results in different formats. A couple of things that I find very helpful. Here's an alignment here between two coding regions. I can see some mismatches there. But what I can't see is the fact that that gap at the beginning actually interrupts the coding region and gives rise to a frame shift. So if I add the CDS feature, and I select pairwise dot for identities, then I can quickly see the differences between these two sequences. And in fact, I can quickly pick up that there is a sequencing error, probably. I happen to pick the alignment between two different species for the same sequences, but you can see that insertion, that relative insertion there that throws a reading frame off. And what I've done here, BLAST has done it for me, is it's translated my query sequence and found the reading frame that matches best. And then it's pulled the coding region from the database sequence, so I can see the changes there. So this is good for highlighting frame shifts and sequence changes. And it gives you the changes at both the nucleotide level and the protein level. Another topic that's very useful for people that are using BLAST on the web, and that's the ability to manage your results. And the request ID is the key to that. And you'll notice when you run a BLAST search, you'll see at the top of your output, there is this string of letters and numbers, and that's a BLAST request ID. If you click on that thing, it will give you a nice URL that will let you retrieve those results. You can send that to us. You can send it to your colleagues if you have an interesting set of results. Those results will stay live on our servers for about 36 hours unless they're enormous, and then we'll have to delete them. But it allows you to share your results and to reformat them. And of course, if you have a problem with your BLAST search, send us that RID, and then we can look and we can see what happened. We can figure out if it was something that you could do differently to make it work better, or if there was a bug in BLAST and we need to fix it. Also, there is a recent results tab on the BLAST page. It allows you to input a BLAST request ID. And as I said, the results are stored there for 36 hours. So you can go back to this page and link there and get your results back. Now, if you happen to close your web browser, then poof, it's gone. However, if you're signed into MyNCBI, it will be there. So that's a really good thing to do because MyNCBI will track your BLAST history. So when you're running a bunch of BLAST searches, make sure you're signed into MyNCBI, and then you can go there. There's even a little pod there in the MyNCBI that will let you go back and get to your BLAST searches. Also, you have the ability to save search strategies. And again, sign into MyNCBI so you can go back to those. And so if there's a way you want to set up a BLAST search to run it again later, you can save that, come back and get it, and run it, say, a month from now to see if anything's changed about that. Finally, in terms of managing results, you can get your results in other formats that are not available as a web page through the download options here. And so you can just expand that section at the top of the page, and you can download your alignments in a bunch of different formats. 3 of them I've highlighted here, the XML and the 2 versions of the hit table. These are nice because they let you parse your results. The XML, in fact, you could basically use that to generate your own display if you wanted to write some code, because it's got a nice tag delimited format that will let you reassemble the entire BLAST output, including the alignments. The hit table is more about, here's my query sequence, here's my hit, here's the score numbers. But it's compatible with Excel and other spreadsheet programs, which a lot of people like. You can also download your search strategies from here, and those can potentially be used again on the web or in standalone BLAST. Okay, and let me mention a few of the specialized BLAST services here. I'm not going to go into them in detail right now because I think we can more profitably visit them live when we go to the web pages. And there are 3 of them that I'm going to have time to talk about today. One of them is Cobalt, and Cobalt is another acronym like BLAST. Cobalt stands for Constraint-Based Alignment Tool. This is NCBI's global multiple alignment tool. It's the only one that we have written. It uses conserved domains to guide the alignment. Almost all of these multiple alignment tools use some kind of strategy to start out your initial pairwise sets and expand that to a multiple sequence alignment. It's an extension of BLAST. That's a useful thing because remember what I said about multiple alignment tools. You need to know that your sequences are related to each other. BLAST will tell you that. You can take a BLAST output and feed it into Cobalt to make a multiple alignment. Primer BLAST is another BLAST variant. It's really a hybrid between Primer 3, which is a sort of a standard kind of primer designing software that deals with things like thermodynamic properties of oligos and things like that, and melting temperatures. And it combines with BLAST so that it can do a specificity check. Not only that, if you use RefSeq as your template sequences. If you put an accession number in there that we recognize, we can use information about exon boundaries and splice variants and where SNPs are to help you design primers that do exactly what you want them to do. And finally, there's MolBlast, which is new. And MolBlast is an acronym that actually doesn't seem to stand for anything, but it's a name that's stuck. So I can't tell you what it actually stands for. But they like the name. They were even going to make the mascot be a star-nosed mole. This is a tool that was designed by our taxonomy group. It's very useful for identifying 16S and other kinds of targeted sequences. Someone in our group has called these things measuring tape sequences. They're sort of these sequences that will let you quickly identify an organism based on that. They're used a lot in things like metagenomic studies. And so for fungi and for bacteria, ribosomal RNA sequences are very important. What it does is it does a blast search and clusters these things with the most similar database sequences, and then draws a tree for you so you can quickly get some idea about what kind of taxonomy or taxonomic units were present in your sample. It also has a feature of being able to tie these things back to sequences from type material so you can get accurate identification. Okay, so I just want to, before we move over to the web pages, I want to just point out to you that Tao and I, who are both here, are part of the Blast Help group. There are 4 of us that answer that email, and we work closely with the development team at Blast, so we're able to take suggestions, to do bug reports, to figure out what's going on with your search and things like that. So you can write to us directly through that email address that's given at the bottom of the slide, blast-help@NCBI, et cetera, et cetera. And then there is this help tab on the Blast page that has access to lots of documentation about Blast. And there's a manual for Blast and other things that are very helpful here. Okay, so what I want to do now is get away from the slides and move over to the web service and do some examples to just point out some features of Blast. So I want to do a sort of a standard kind of a Blast P search with a human protein, a creatine kinase. And I want to show you Cobalt with that. So we're going to make a little protein family tree of the creatine kinases from mammals. Then I want to show you some things with Genome Blast. So this is our BLAST-N example, really. We're going to take a messenger RNA from tomato, find the corresponding gene in the potato genome, and then find that region in the potato genome and then do an SRA blast with that. Because a lot of our potato genes are annotated by using alignments from RNA-seq data. And so we can sort of replicate that ourselves by searching a set of data from potato. And then we're going to use Primer Blast to design some specific primers for an exon of BRCA1. Then we're going to do a Microbial Genomes Blast example, looking for the identification of something that we found in the chicken gut metagenome. And then finally, I'll show you MolBlast quickly just to show you how it works, and we'll take some sequences from the cow rumen metagenome to do that. Okay, so I'm going to leave the slide set now, and I'm going to go over here to the NCBI homepage. And as I said, the materials are on our FTP site, and there'll be a link to those that we'll have available later on the webinars page. But I may have to refer back to this sheet that's in the FTP site. It has the RIDs. Some of the blast searches that I'm proposing to do take a long time, probably longer than we want to wait. And so there may be cases where I can set them up, but I need to go back to this sheet and sort of get the RID and then retrieve the results. And then I'm using the fact that these RIDs are stable. Now I have a trick that I can use. I can make these last longer than 36 hours. But even for you, 36 hours is quite a while. If you ever have to give a talk about BLAST, you can always get the RIDs, and if you do them the day before your course or your talk, they'll still be available. So the first thing I want to do is to start with the human protein. I'm actually going to show you a way into BLAST through the NCBI sequence databases. And so what I'm going to do is retrieve that protein from the protein database. So I'm going to change my pull-down list here on the NCBI homepage, and that's easy enough to find. I'm sure that most people listening to this webinar have been there before, so this is Just search Google for NCBI and you'll find this page. And I'm actually going to look for a particular gene. We have a nice little shortcut that's pretty handy now. So if I just type a gene name like that, CKM, creatine kinase M type, which is the muscle creatine kinase, we have this gene sensor that detects that that's a gene name or a gene symbol, and it automatically will find for me the human protein. So, I mean, I can go through all this stuff here. There's 264 protein sequences, but really what I want is this one. And so there is my sequence. You may be familiar with these proteins. They're involved in rapid generation of ATP and things like muscle cells. And there are actually 4 different genes in mammals. And so what we can do is sort of make a little protein tree with this example. The other thing about this protein is that it has a conserved domain that's present way back. So it goes all the way back into bacteria. And so we can see that with BLAST if we want to. But this example sort of points out to you how big the protein databases are now and why it's very important to limit your search to exactly what you want to find. So if I want to run a protein BLAST search with this, I can just click this link over here, which will basically just set up the BLAST page for me. And so all it does is it puts the accession number into that query box. And of course, if I have my own sequence, I can put that in there in FASTA format, but I don't need to in this case. The accession number will do the job. And what I'm going to do is to set this up to run against NR. If you try this at home or wherever you are, you'll find that this is a really huge search. In fact, to make it even tractable, I've got to exclude the models that we've generated from the vertebrate genomes because our genome annotation pipeline has been very busy. And I've also got to exclude some of the uncultured bacterial sequences that are in here. Because as I said, the conserved domain of this protein is conserved all the way back, and so we'll see bacteria in here. In fact, it's conserved very far back, and so what I have to do to make this even work so that I can see that far back is to do what I said earlier and change the maximum target sequences. And actually, I need to change it in this case to 5,000, in fact more really if I want to go all the way to an e-value of 10. I'm going to leave that at 10 just so you can see what happens when you do that kind of a search. Now, I'm not going to actually run this search. I mean, I can press the button, but it takes a while, this one in particular, because I'm searching the entire NR database, and it's going to take a while. Now, one of the things that I do want to point out to you is the results from the conserved domain database search, which remember is reverse Sra Blast, they're available almost immediately. And if I didn't know what this protein was, if this was some unknown protein from an annotation pipeline, I already, right here, I know that this is a creatine kinase-like protein. It's probably a mammalian creatine kinase. And I could click through that to the conserved domain database to quickly identify the kind of protein it is and its potential function. If I'm interested in finding out what the homologous proteins are, I have to get my BLAST results. Now, what I can do is go over here to the Recent Results tab, and notice it shows me my running BLAST job here. But I have saved these set of results so we can look at them just by entering the RID, and so that's what I wanted to show you here. So this is the sort of no-holds-barred kind of BLAST p-search. I'm just going to copy that and go over here and put it into the request ID box and then retrieve my results. So this is the formatting page. So I have the opportunity here to sort of change the way this is formatted. I asked for 5,000 descriptions. That's what I'm going to get. The graphical overview, I can change that if I want to. It's not terribly informative in this case, so I'm not going to bother with it. We'll see one in a few minutes. So I'll go ahead and view report. And these results take a while to load typically because they're so large. Notice that it shows you the way that it managed those filters to get rid of the models and to get rid of the environmental samples. Here, there's my request ID. There's my conserved domain database results. My BLAST graphic here is not terribly useful because it's maxed out at 100. I didn't set it to anything higher. And here are my hits. So I have a self hit. This is the protein I started with. There's a bunch of different things here. And my e-values the main thing that I'm interested in really is over here. This tells me about the significance of my matches. So notice that we're starting at an e-value of 0. What does that mean? It just means that the e-value is so small that we didn't bother to report it to you. It doesn't mean anything special about that alignment. Some of these are 100% identical, but not all of them that have e-values of 0. And then in order to find what I want to do is to get down to the bottom of this to show you what it looks like when you actually get to the place where you have alignments that are not distinguishable from chance. So you have to go a long ways to get there. But this is a really good example just to show you what it's like if you search in R without any kind of restrictions. And so at the very bottom of my output, I've stopped at an e-value of 1.5. I probably would have had to ask for 10,000 descriptions to see all the way to an e-value of 10. And some of these are fragmentary proteins, but a lot of them are actually, some of them are related, potentially related proteins. They may be fragments because they say things like guanidinophosphorotransferases, if that's actually something that has the same conserved domain as creatine kinase. But what can I tell about these hits? Nothing. If I have an e-value of 1, it means I cannot distinguish my results from chance. Where that exact cutoff is, where you decide that something's not due to chance, is a matter of taste and a matter of how would you feel about being wrong. So that's why there isn't a strict cutoff here. And we can take a look at one of these alignments if we want to. I think I'll show you one from the next set, which is a little bit prettier and cleaner to look at an alignment. So what I can do to manage my BLAST search is, if I don't like what happened when I ran a search, all I have to do is hit edit and resubmit. And what I'm going to do here now is to change the database, which is an important thing to do. And I can change it to a subset of NR, and I'm going to change it to our reference sequence protein set, a smaller set of data. And I can make this data even smaller if I want to. Say I'm interested in collecting these for particular organisms. So notice that when I start to type mammals, I get the corresponding taxonomic identifier here. I can select that. I'm going to leave these limits on like I had them before, so that I'm not interested in the model sequences. This uncultured environmental sample sequences really doesn't apply in this case because I'm working with vertebrates, but you can leave that on. It doesn't much matter. Then I'm going down into the algorithm parameters here. I know from experience that I'm not going to get that many proteins, so I can change that back to 100. The most important thing, though, I can do is I can use the e-value here and change it to something more stringent. And as I said, what you decide is more stringent is a matter of taste. I'm just going to set it to 1 times 10 to the minus 30th, pretty much arbitrarily. But you can play around with this to see what works well for you. And then I can run the search again. This one typically doesn't take too long, so let's see how slow it is. If it's very slow, I won't suffer along with it, and I'll go ahead and retrieve the results. And how long it takes depends upon a lot of things, like how busy we are at NCBI, what set of servers you wound up running it on, and things like that. But I don't want to wait, so let me go ahead and go over here to the recent results, and I can again use my request ID to retrieve these results. So I have it over here in my document. Copy that. And so again, another request ID. And now the set of results is much cleaner. My graphical overview is actually kind of useful. I can see there basically seem to be 2 different kinds of proteins, ones that align the full length and ones that are missing the N-terminal and C-terminal alignments. And if we look at our descriptions down here, we can see there are several different types. These are different gene products, actually, the M type, the B type. These are the cytosolic isoforms. And then the S type and the U type are also nuclear genes, but they're targeted to the mitochondrial, so they have a leader peptide on them, and that makes them not align at that end. You can read the organisms here. These are things you probably recognize. There's olive baboon, rabbit, sperm whale, which is an interesting one. And I can pick any one I want to look at the alignment if I want to. So let's just say this S-type mitochondrial precursor from mouse. If I click on this link on the left-hand side, it jumps me down there. And so you can see the way BLAST renders that alignment. Notice that it's a local alignment, so not all residues are included. It doesn't align at the very beginning, and it doesn't align at the very end. Another point that I should make is we mentioned that BLAST searches a non-redundant protein database. In this particular case, the mouse and the rat protein happen to be identical, and so they're both represented here by one sequence. But you can see that when you expand the titles that there are two different proteins here represented. They have exactly the same sequence. What would be useful to do now that I have this set of sequences is let's make a global alignment out of them. And in order to do that, we can use Cobalt. And the way to get the Cobalt is up here. There's a set of links here that says multiple alignment. And this one doesn't take terribly long, so let's let it do its thing for a couple of seconds. And you can see a little warning that Cobalt gives me. It says all queries form only one cluster. And that's because these proteins are so similar to each other in terms of their domain structure that it wasn't able to use that shortcut. You can go back and use the edit resubmit link here to adjust this to do what it suggests and turn off query clustering to see if it affects the alignment. In my experience, it doesn't in this particular case. And then here is your multiple sequence alignment. It includes every residue of all the proteins that we sent it. We can change the way that's formatted here if you want to see the complete alignment. It's plain text here. And you can see that leader peptide down here that didn't get aligned to the cytosolic isoforms. And of course, it's a matter of taste what you do with those gaps and things like that, and that's beyond the scope of what BLAST can do for you. BLAST can certainly let you download this alignment from the downloading options up here. And this is the download tab here. You can get this in various formats. You can also get a nice little phylogenetic tree. We have a Treeview program that will let you look at the distances between these proteins. That's kind of nice in this case. You can see the clusters, the mitochondrial genes, targeted genes, and the cytosolic targeted genes. The collapse mode here is BLAST name. I can change it to show all. And you can see a nice little tree there. And again, you can download this tree if you want to and manipulate it locally. There's a download option available from the Treeview here. Okay, so the next thing I want to do is to do a BLAST in search. In this case, it's going to be a search against the potato genome. So I'm going to go back. I want to close some of my tabs here so I don't get too confused. I'm going to go back to the NCBI homepage. And I can take advantage of that gene sensor again. And I can go to nucleotide and I can type in, I'm interested in this thing called the ethylene receptor, ETR2. And it just so happens that this is set up in such a way that it's fairly convenient. So if I look at my transcript sequence here, I have the accession number of the tomato ethylene receptor. So I'm just going to copy that to my clipboard. And then I'm going to go back to the NCBI homepage to get to BLAST. Just open that in a new tab and go to BLAST. And what I want to do now is to search a particular organism's genome, and I'm going to search potato. So I can search the reference assembly for potato here. I can also search other products or things that have gone into our annotation pipeline, but I'm going to go ahead and leave that set the way that it is. And then I can just paste my accession number in there. And I'm going to run this as a megablast search. Now, you might stop me if you were paying attention because this is a cross-species comparison. Tomato and potato are fairly closely related organisms. They're in the same genus by some people's estimation anyway. So I'm going to run that, and that typically doesn't take long. Megablast is faster than BlastN. So I have three sort of distinct kind of hits. Theirs are aligned onto my messenger RNA. This little black line here lets you know that these hits are separated on the genomic sequence by some distance. And so basically what I've done is I've highlighted the exons of a gene. I've got 85% of my query covered, so I didn't align all of it. 97% identity over the highest, that's the highest scoring region. And I can look at my alignment down here. And that's not terribly interesting. What would be helpful in this case is to do that modification so I can see where the coding regions are and things like that. So I can go up here to the formatting options. I can change the alignment view to pairize with dots for identities if I want to, and I can add the coding region feature on there. And that just went back and reformatted my results. And so I can see that the tomato and the potato are quite similar. And basically these are the exons of the potato gene, highlighted by BLAST. Now BLAST doesn't know where to put the splice junction, so sometimes the alignment, this is pulling my coding region from the predicted gene that's present in the potato genome. And so you can see this little squiggly line here is saying that that's not where the splice junction ought to be. But that's okay. It does a pretty good job of highlighting the exons. This particular case, these are in exon order. Sometimes they're not. By default, they're ordered by score, by the e-value really. Just arbitrarily, this happens to be the order that the exons are in. So if you change it to query start position, for example, it doesn't do anything. The query start is right here. Now notice we're missing the first 65 bases of this. It's interesting to take a look at this in the graphical view on the genome. And that's another thing you can do with these Genome Blast pages, is I can take a look at this super contig here that represents this genomic scaffold from potato. And I can go to the graphics view here. And I can see well, there's one of the exons of that gene. You can see where there are mismatches. Let me zoom out a little bit. And so you can see quite clearly when you look at the structure of the gene here, you can see the exon intron structure of the potato gene. My tomato messenger RNA sequence highlighted most of it. We actually missed a small upstream exon that's right there. I will leave it as an exercise for you to go back and do, and to try this with BLAST-N. What you'll find is that you pick up that upstream exon. You also pick up some additional alignments for the other paralogs. There's more than one ethylene receptor gene in the potato genome, and so you find the other ones as well, and you find more of them individually and better alignments. So megablast is quick, but it's not terribly sensitive. Now, the next part of this is to really do something with the Sra Blast, just so you can see how that works. And so what I'm going to do is I'm going to put my mouse pointer over this gene object here, and I'm going to use this as a way to get to the genomic coordinates of this particular gene. So I'm going to go to the gene ID here. This is going to leave this viewer here, and I'm going to go to the gene database. So we already did know about this gene. It says ethylene receptor 2-like because we're not terribly confident about it. But notice that I now have the coordinates of this gene on that scaffold. So I can use this as a query to sort of look at the RNA-seq data. And that's what we did here. You can see how we have used this. See these, this histogram here shows you where these RNA-seqs align to the genome. And that's basically how we called this gene, is based on the alignments of this RNA-seq data. So just to show you Sra Blast, we can sort of reproduce that ourselves, take this genomic region and search it in SRA. In order to do that, I'm going to need to find a particular set of SRA data. So let me go ahead and do that. So I'm going to leave this open over here. I'm going to go back here, and this is a little bit of a departure from BLAST, but it won't be too much of one. So let me go to a different database. And this is the database bioproject where you would go to find things like, sequencing projects and things like that. And it's a good place to find the next-gen projects. And so I'm going to type in here a standard kind of query at NCBI, which is I'm going to do an organism search for potato. So I have 78 records, and notice I have these nice little filters over here, and I'm interested in transcriptome data sets, so that would be RNA-seq data, and then I'm interested in ones that have project data and SRA. And there are lots of different ones here. One that I'm going to pick just arbitrarily is this one from the University of Minnesota. That's number 10 on my list. This is just a way of getting to the SRA experiments. They were interested in the effect of potato blight on gene expression. So I can go ahead and link from here to SRA. And there are 36 of these experiments. It's quite a tall order to search all of them at once, so I could just pick arbitrarily a short set just to show you how this works. I'm going to pick the first 5. And then I can use the send to menu here in SRA to send these to BLAST. Now this does open a new window depending upon how your web browser is configured. There it is. So now I've got these 5 SRX databases here. I could also come here and start typing any kind of a name for these things. This is the Sra Blast interface. If I know the SRX accession number, or I know something about the organism, like I could start to type potato, I would get a lot of experiments. So in order to use the SRA database on the pull-down list on the BLAST page, you would have to know the name of the experiments. So now what I can do is, just to show you a way to use BLAST where you can limit the subrange here, I can take the potato accession here, and I'm just going to grab this whole string right now. I'll show you what I'm going to do in a minute. So this is my accession number here. What I want to do is to use these ranges here on the query subrange on the right-hand side. So I can run this search, and I'll leave it at MegaBlast for now. These are short reads, and if you're trying to go across species or something like that, you might need to go to BLAST M. I'm going to go ahead and run that. This will take a while to run, so I'm not going to wait for that. I'm going to go ahead and retrieve the corresponding result that I saved. So I can go up here to my recent results. And I can retrieve this RAD here. And this is a limited set of alignments here because this is only showing me 100. It's useful to, on these, to often change your formatting options. So in this particular case, I asked for 1,000. So it's worth seeing that we're going to get a lot of them. And that may take a minute to download. Let's see what happens. But then you can see that these alignments show up and they're right along the places where the exons are. And that's essentially how we annotate these exons. We use a tool that takes care of the splicing, but still you can do a good job of highlighting the exons that way. And that's what the RNA-seq data are like at NCBI. You can search them using this BLAST tool. Sometimes it doesn't like it if I put 1,000 on the graphical overview. There we go. And these are paired reads, which is kind of nice, and you can sort of pick that up from the way the BLAST graphic is here. So these are the different exons. The tall ones are the ones that we call as exons. These other ones are probably data that needed to be filtered out before we call the exons. These are all available from SRA. So if you click here, instead of going to, you go to the alignment here, and then if you want to get to the sequences, you're going to click here to go to the SRA run browser. You can look at that particular read. That's got a pair. So there's one of these is one of the ones we hit. The other one is probably in our output somewhere, and you can download that data from here. Okay. The next thing I want to do is the BRCA1 example with Primer Blast. So I'm going to close this one and this one as well. And I'll go back up here and get an NCBI homepage. This is a real common application for a tool like Primer Blast. And what we want to do is to design a set of primers that work on a particular region of BRCA1. So I'm going to click here, run the search against nucleotide to retrieve the sequence of the gene that I want. And again, this is set up to give me exactly what I want. I can get a genomic sequence here. That genomic sequence is our Rps gene record, which is a fully annotated fixed record for that gene, so it has the exon sequences in it, things like that. I can retrieve that here. And then I want to find the sequence of an exon. So this is, again, a feature of the sequence database, but it's pretty handy. I can click this link that says Highlight Sequence Features here. And I've got various kinds of features. The one that I'm interested in is exon. So I can just skip ahead until I find exon 15. And exon 15 is a place that does contain some disease-causing changes in the BRCA1 gene. I can display this in FASTA format. And now I have it in the sequence database, and I can go ahead and submit this to make the pick primers. If I click the pick primers link here, this is exactly the same idea as running BLAST, but now I'm going to run Primer Blast on this nucleotide sequence. So now I'm in the Primer Blast interface. There are two ways to use this interface, two main ways. One is you have a template sequence, which I've given you here, and you want to find primers that are specific to that template sequence. The other one is I have a primer, and I want to see where it binds, or two primers, and I want to see what they would amplify. So what I'm trying to do is to amplify this exon. So a useful thing to be able to do is to change this so that my primer binding site is upstream of these things. So what I can do is take my forward primer sequence and move it and back up. So at 146, instead of 146, 746, I can do 146, 646. And the reverse primer, I have the opposite problem. So I need to go from the end of that out 100 bases. So I'm going to add 100 bases to the end of that. Now, I'm going to go ahead and run this. I need to figure out what my background database is. So what I want to do is to amplify this from the genome. So notice that the default is to use refseq RNA. I'm going to use reference assembly. The organism here is given you as the tax ID. I just want to show you that this really is the same as picking human. There are lots of other things I can do here. I could tell it not to find primers that land on a SNP. I could adjust the range a little bit more. You'll see that we'll wind up being inside the exon just a little bit, but we could move it if we wanted to. I'm going to run this. I'm going to let this run to completion because there isn't any good way to get back the primer blast RID. Now notice that it realized that the sequence I'm searching with is not actually in the database I chose, but it says here's something that you might be interested in. This is chromosome 17, and that's right. That's where BRCA1 lives in the human genome, so I can just say that's fine. And then it's going to design primers, and then it's going to check those primers to make sure that they don't amplify anything else in the database. And if they do, it's going to tell me about potentially what the mispriming sites are, how many mismatches there are, and things like that. So we'll let that run for probably take a minute or two for that to come back with a result. Okay, and so it found 3 sets of primers for me, and it's got a nice little graphic here that shows me exon 15, and there's where my primers would bind, and so it would amplify this region of BRCA1. I could add the Snip Track to this view to see where the potential SNPs are. So this is the kind of thing that a lot of companies that sequence DNA for clinical studies would do. They would amplify a particular exon of a gene to look for changes. And here's my set of primers. I could happily go off and order these from whoever supplies my primers, manufacturer, and they should probably work. And if you need more primers, you can ask for more. These were it typically gives you 5 sets. Probably the next set would not be as good as these in this particular case. Okay, we have time, I think, to do really only one more example. There are 2 that are sort of possibilities that you can look at. I want to make sure that we get to see Mole Blast. I think the first one I want to do is to go ahead and search another Genomic Blast page just so you can see what a WGS hit looks like. These are whole genome shotgun records, and this is an unassembled genome that we can search. And this is the chicken gut metagenome. So let me go ahead and go back to the Blast page here. So I'm going to go back to the NCBI homepage, and I'm going to go back to Blast. What I'm going to do here is to go ahead and type in a metagenomic name here. So this is chicken gut metagenome. If I click here, I'm going to get a Blast page for that metagenome. And you can see that what this is basically a whole bunch of WGS contigs. They're not assembled, and that makes sense because these represent multiple organisms. In some cases, people make scaffolds out of these kinds of things. And what I'm going to do just to show you is a particular kind of sequence that's useful, and that's one of these 16S sequences from one of our targeted loci projects. And just to save us some time, I'm going to go ahead and just use the accession number, which is NR043437. And that's a 16S sequence from a Bifidobacterium longum genome. That's designed to be essentially a reagent for identifying bacteria, if you will. Bifidobacteria or actinobacteria that wind up being in things like guts. Chicken guts, human guts in particular. But let's see if we can find anything that matches this in the chicken gut metagenome. So I'm going to go ahead and run this search, and I can retrieve it too in case this turns out to be a long search. So I just wanted to show you this so you can see that what happens when you hit a WGS style sequence. It'll be a slightly different kind of result when you get to the sequence itself. So I'll go ahead and retrieve this using the recent results like we've done before. So we have lots of matches to that 16X sequence. The best one is this one here. And I just want to show you something that you may find is unexpected. This sequence, of course, would only be in the whole genome shotgun sequence database if you searched it on the BLAST homepage. This is a much more convenient way of doing that. We have to treat the WGS sequences differently than we treat other sequences. They're not really a part of the Entree system directly. So if I retrieve the succession number, you may be surprised to find out you're in a sort of an environment that looks a little bit more like SRA, and that's in fact kind of what this is. So if I wanted to do more with this sequence, I would basically have to click here to get the FASTA format. And as an exercise, you can try searching this on another Genomic Blast page that I wanted to show you today, but we probably don't have time, and that's the Microbial Genomes Blast page. You can try to find a corresponding microbial genome that matches this particular section from the chicken gut metagenome. And it definitely looks like it's in the Bifidobacterium genus, but I don't think we actually have a genome that corresponds to this one. Last thing I want to show you, and then we'll have time for a couple of questions, is the new service that we call MoleBlast. So in order to use MoleBlast, I need to get a set of sequences, and I'm going to have to resort to BioProject again to do that. So I'm going to go back to the NCBI homepage and pick BioProject, and these are going to be ones from the cow. So I'm going to use a bovine gut metagenome. Now I have some things turned on here that I need to turn off. I need to turn off Transcriptome and SRA because I'm not just interested in that. Let me run the search again. And I'll pick the ones that have links to nucleotide. And I'll use this one. This is the pooled gut metagenome from STEER. These actual individual STEER metagenomes are available too. I'm going to click here. And I'm going to go ahead and get these pieces of genomic DNA. And they all say that they're uncultured bacterial clones, and so we might like to know, what are these exactly? So I'm just going to take the first 20 of them just so you can see how this works. And what I can do to get some things to work with here is to get an accession list. And there are 20 accession numbers here that I can go ahead and copy to my clipboard. Get the last one. And now I want to go back to the BLAST homepage. And I want to select MoleBlast, which is available down here in the bottom. And I'll go ahead and paste those in as queries. Now, MoleBlast lets me cluster these using BLAST, and then it will create a tree for me. It will also create a multiple alignment for me. But the useful thing to do it here is to look at the 16S ribosomal RNA sequences. That's what this project had in it, were these targeted sequences from this metagenome. So this might give me some useful information about what classes of organisms are in there. This is a common kind of thing that people do with metagenomic studies, is they talk about operational taxonomic units. So we could think about, what bacterial phyla are represented, for example. There are advanced parameters that you can play with down here if you want to. I'm just going to use the default ones right now, and I can run that as an alignment. A MoleBlast is not terribly fast, so I think it again might be useful to retrieve this as an RID. The MoleBlast RID system is separate from the BLAST RID system, so the recent results tab here is going to be different than what we have for BLAST, but I do have the MoleBlast RID right here. So this is the Treeview display that you're used for. And it's classified things for me. The ones that say bacteria, the blue ones, the green dots over here represent my query sequence. So these are the 20 sequences that I submitted. And you can see what ones they come out closest to. So for example, I would say that we probably have one in the genus Blautia here. Other ones, it may not be so clear what genera are present. But for example, if you're interested in knowing about higher bacteria classes or perhaps phyla that are in here, these are the firmicutes are the ones that are in blue here. And the CFB group, the things that some people call bacteroidetes, are in brown. So this would be a way of getting sort of an idea of what bacterial phyla are represented. And I could do this with a lot more than 20 sequences. I just was trying to show you a small set. And you can see the alignment by clicking on this link here and download the tree in various formats. So I think we should stop here. I think we have about 5 minutes to sort of look at a few questions that people have. I think we probably see a lot of activity in the question pod. Tao answered most of the questions in writing. He was busy typing the whole time. Okay, so Tao already sent these questions to everybody. So I think what we'll do is wrap it up, and I will go ahead and make these questions and their answers available to everybody in a document. And that'll probably take me about a week to get it collated and up on the site. And these things will all be linked to our webinars page. Let me just show you where that is. I'm going to go back here to the NCBI homepage. And you probably have noticed that we've changed the homepage, and there is now this Learn page here. There will be a Webinars and Courses page here. And so the one that you're attending right now is going to be available over here on Archive Webinars and Courses. And I will link the materials and eventually the recording of this to that page. By the way, if anybody wants to, or if you may have learned enough about Mole Blast today, but I have a webinar next week at 9:45 a.m. where we just basically introduce it. There won't be a whole lot more detail on that, but you might pass that around to people that didn't see this and are interested in Mole Blast. That's a new thing called the NCBI Minute, where we do a quick introduction to a new NCBI tool. Okay, well, I think we're going to conclude the session for now. And you can please write to us at webinars@ncbi.nlm.nih.gov. You can also write to Blast Help with any questions specifically about Blast. Okay, I'm going to end the webinar now. Thank you for coming.